Welcome to the ultimate guide to Mendelian genetics. Have you ever wondered why you have your mom's eyes or your dad's smile? The answers lie in the principles of inheritance and variation. Today, we are breaking down how traits are passed from parents to offspring, a study that forms the very bedrock of modern biology. We'll explore the paradigm-shifting work of Gregor Mendel, who cracked the code of heredity using simple garden peas. Whether you're studying for an exam or just curious about how life works, this step-by-step breakdown will make genetics crystal clear. Let's dive in. Before we get into the science, let's establish some foundational terms. Inheritance is the process by which characters are passed from parent to progeny. It's the absolute basis of heredity. Variation, on the other hand, is the degree by which offspring differ from their parents. Interestingly, humans exploited these principles long before knowing the actual science. Between 8000 and 1000 BC, our ancestors used artificial selection on wild populations to breed desirable traits. A famous historical example of this trait exploitation is the Indian Sahiwal cow, bred for high milk yield through the selective breeding of wild cattle. For centuries, heredity remained a mystery until Gregor Mendel came along. Conducting hybridization experiments on garden peas from 1856 to 1863, Mendel achieved a massive paradigm shift. He succeeded where others failed, because he was the first to apply statistical analysis and mathematical logic to biological problems. His success relied on three crucial pillars. First, a large sampling size, which gave his data immense credibility. Second, successive generation testing to prove general rules rather than isolated ideas. And third, his use of true breeding lines, which showed stable trait inheritance over several generations. To make his experiments precise, Mendel selected 7 distinct, contrasting pairs of traits in garden peas. These experimental parameters included stem height, which could be tall or dwarf, flower color, either violet or white, flower position, axial or terminal, pod shape, inflated or constricted, pod color, green or yellow, seed shape, round or wrinkled, and seed color, yellow or green. By utilizing true breeding lines that express stable traits continuously over generations through self-pollination, Mendel established a controlled baseline that eliminated unpredictable variables from his data. Let's look at Mendel's first big experiment, the monohybrid cross. Mendel took a true breeding tall plant and crossed it with a true breeding dwarf plant. This initial pair is the parental generation. When he raised the seeds, the resulting offspring, called the F1 or filial 1 generation, turned out to be 100% tall plants. This was a shocking observation at the time. There was absolutely no blending of traits. The dwarf characteristic had completely disappeared from view, and the progeny looked exactly like just one of the parents. Where did the dwarf trait go? Mendel didn't stop there. He took those F1 tall plants and allowed them to self-pollinate to produce the F2 or filial 2 generation. This is where the big surprise happened. The dwarf trait suddenly re-emerged, and it came back with no blending whatsoever. When Mendel counted the plants, exactly 1/4 of the F2 generation turned out to be dwarf, while 3/4 remained tall. This gave a precise 3:1 phenotypic ratio. This proved that the dwarf trait wasn't lost or destroyed in the F1 generation. It was simply hidden, waiting for the right moment to show up again. To explain these results, we need to learn the lexicon of life. Mendel called the units of inheritance, quote, factors, which we now know as genes. Slightly different forms of the same gene are called alleles. If an organism has identical alleles, like capital TT or lowercase tt, it is homozygous. If the alleles are dissimilar, like capital T lowercase t, it is heterozygous. In a heterozygous pair, the dominant allele expresses itself, while the recessive allele remains hidden. This explains why genotype, the actual genetic makeup, differs from phenotype, the physical look. Now let's examine the mechanics behind this, the law of segregation. During gamete production, or meiosis, the two alleles of a parental pair segregate from one another. This segregation is completely random, acting like a fair coin toss. There is exactly a 50% chance of a gamete receiving either allele. For instance, a heterozygous parent with capital T lowercase t alleles will produce equal proportions of gametes carrying the capital T allele and gametes carrying the lowercase t allele. This absolute randomness in segregation is what drives the predictable patterns of heredity. To visualize this probability engine, we use a diagnostic grid called the Punnett Square. By crossing the gametes of the F1 generation, we can see exactly how the F2 generation forms. Mathematically, this random fertilization is identical to expanding the binomial expression, half t plus half t squared. The Punnett Square reveals that the 3:1 phenotypic ratio of tall to dwarf plants actually masks a precise genotypic ratio of 1 homozygous tall to 2 heterozygous tall to 1 homozygous dwarf plant. Math and biology align perfectly to explain the mystery of life. But how can we actually tell if a tall F2 plant is homozygous dominant or heterozygous? Since they look identical, scientists use a diagnostic tool called the test cross. We take the unknown tall plant and cross it with a known homozygous recessive dwarf parent. The offspring tell the story. If 100% of the offspring show the dominant tall trait, the unknown parent was homozygous dominant. But if the offspring turn out to be a 50-50 mix, the unknown parent was heterozygous. And that is the elegant, predictable world of Mendelian genetics. And there you have it. We have journeyed through the foundational principles of inheritance and variation, uncovered Mendel's historic breakthrough with garden peas, and demystified the mechanics of monohybrid crosses, Punnett squares, and test crosses. Mastering these core principles is your ultimate key to unlocking more complex genetic concepts down the road. If this step-by-step guide made genetics click for you, make sure to hit that like button, subscribe to the channel, and turn on the notification bell so you never miss a lesson. Drop a comment below if you have any questions, or let us know what biology topic you want us to break down next. Thanks for watching, and we will see you in the next video.